we'll have 2 antiparallel strands. That means that here we'll have guanine, and that will always bind to a cytosine. So G's goes with C's, and then A's go with T's. And this also has to do with how they're made. So enzymes will actually break apart these strands. That's the only time we see single-stranded DNA, when they're being broken apart for synthesis. And every time it sees an A in one strand, it will actually take a T, and the enzyme RNA polymerase will bind that T to the A. And only T's can bind to A's, and only C's can bind to G's in a chemically stable way. And that's because one of the pairs have double bonds and one of the pairs have triple bonds. Now these bonding pairs that make up these double helix, so let's just fill it out. So let's say A, T, just give it a random code, A. So just imagine we have this big double helix, all winding, and we have all the base pairs on one side, and the enzymes have done their job, and we have complementary base pairs on the other side. What is the difference between this and RNA? Well, as many of you already know, RNA is only a single strand. That's important. And so we have G, T, C. But one important difference is we have a U. And this is actually how your body can tell whether it's dealing with DNA or RNA. If there's little bits of DNA floating around your body, then your body will actually destroy it, because your DNA is inside of the nucleus. DNA is kind of like the master copy of your genetic information. RNA, DNA is used by proteins to make RNA. That's what's actually being sent around your body, telling your body what proteins to make. So we see that RNA has uracil, or DNA does not. So that's how your body tells if it's RNA or DNA. DNA comes in a double helix, as we said. And the last thing we need to know is that DNA has a 5' end and a 3' end, and the double structure is actually anti-parallel. Let me show you a picture of what that looks like, because unlike proteins, amino acids, these other ones, you actually do need to know the details of the chemistry of nucleic acids. So if we look at the molecular structure of DNA, oh, that's a video. I don't want the video, I want the article. Sorry about that. So if you look at nucleic acids, there's a very nice picture somewhere here. Sorry for not having this pulled up. Okay, so here's what you need to know about the DNA double helix. So it's made out of these backbones right here that have a phosphorus, a phosphate group. So that's phosphorus bonded to 4 oxygens, and then it's bonded to a carbon chain. And then we always have this this 5 carbon group, and then it repeats. So this is the backbone of DNA. It always has PO4 bonded to a carbon chain, and then this is just a repeating structure. So if you look at this part right here, this repeats here and just keeps repeating all the way down. And these are all covalently bonded to our A's, T's, G's, and C's. And that's these chemicals here. They're not actual letters. Of course, they represent chemicals. A, G, C, and T are all of these. So this is called the backbone, the phosphate sugar backbone. So this is the phosphate part. That's why phosphorus is essential for DNA. Also, you'll notice that nitrogens are essential, oxygens are essential, carbons are essential. So we're using almost all of those essential elements that we talked about back in video 1. Now, the only difference between the backbone of DNA and RNA, and this one you do need to know, is the sugar chain, saccharide chain, that is in DNA is a deoxyribose, meaning this is a H. And in RNA, the structure is ribose, meaning that's an OH. Everything else is the same. It's going to just look like a big strand of a single-stranded. So if we have a single strand of DNA, single strand of RNA, the only way you're going to tell the difference is instead of having an H here, you're going to have an OH. One of them has an extra oxygen. One of them has been deoxygenated. They both have phosphate, these phosphate group. They both have the chain. They both have the bases. Let's see if there's a nice picture of RNA before we go. Yeah, I don't see it because this is about DNA. But yeah, that's all you need to know. So hopefully that's a helpful introduction. Sorry I didn't have everything perfectly pulled up. But what are the important things you're supposed to take away? DNA is double-stranded. It's anti-parallel. It goes from 5' to 3'. So on one side, we have the phosphorus sticking out. On the other side, we have the carbon chain at the edge. And in DNA, we have the base pairs adenine, thymine, guanine, and cytosine. Whereas in RNA,